Well, welcome everyone. Advanced Analytical is pleased to be a sponsor not only at the second Festival of Genomics, but also the first Festival of Genomics, most recently in Boston. So what I'd like to do today is talk a little bit about our instrument, the Fragment Analyzer, and its features and benefits. The Fragment Analyzer is the premier instrument for automating the analysis of nucleic acid fragment and smears. It was released in 2012 and developed to address many of the key bottlenecks that slow result generation in your laboratories. It is a multi-channel capillary electrophoresis-based instrument with unique design features to improve workflow and change how you're doing your QC for your fragments and smears. It is an instrument that automates and simplifies fragment analysis. And what I'd like to do is describe some of the steps in which our instrument works. First, reagents are automatically delivered into our capillaries using this high-quality syringe pump that I'm showing down here. What we do is we pressurize a reservoir right above it, and that pushes the reagents into the capillaries, eliminating the manual addition of gel. Once the nucleic acids, the gel is loaded into the capillaries, the nucleic acids are precisely and consistently injected using a highly controlled process called electrokinetic injection. On the Fragment Analyzer, both the time and the voltage of the electrokinetic injection can be controlled if desired. Once the separation starts, every sample runs through its own capillary, meaning that no one sample will ever affect the run of another sample. This is unlike lab-on-the-chip methods, where a previous sample could affect another sample simply because there's only one separation column. Now, as the nucleic acids move through the capillaries, they pick up an intercalation dye, and then upon reaching the window part of the capillary, we hit it with a powerful LED that excites the fluorescent signal that is detected by a CCD detector that continuously monitors for the fluorescent events. Most importantly, because there is sufficient time for the dye to interact with the nucleic acids, we have better sizing and better quantification due to that resonance time and the interaction of the dye and the nucleic acids. Because of our unique design features, our instrument is the premier instrument for automating fragment analysis. I've listed here some of those unique design features, and I'd just like to cover a few of them in my time. Our capillary arrays come in 6 different configurations. We have a 12 capillary array, a 48, and a 96 capillary array, which are interchangeable by the user. We also have different lengths for our capillary arrays. We have a 22, a 33, and a 55 centimeter capillary array. Their configurations and lengths are listed here. All of our arrays go through a highly controlled quality testing process before they're released for use. One of the hallmarks of our instrument is the ability to switch between different gel matrices. Our instrument is the only instrument on the market that can actually hold 2 different gels, meaning different sample types can be separated, and you can easily switch between different applications, all by virtue of having those gels on our instrument. Now, all of the gels can run through all of our capillaries, so there is no need to switch the capillaries between runs. In fact, our arrays require no special daily handling or maintenance, unlike some of the lab-on-the-chip instruments. The ability to accommodate more than one 96-well plate. Our instrument is the only instrument that holds more than one 96-well plate. In fact, we can hold 3 96-well plates as indicated by these arrows. Sample drawers are always accessible, whether you're loading just 12 more samples or whether you're loading a whole plate of 96 samples. Diluted samples can be re-injected into the capillaries if desired. And when you include how many gels we can put on the instrument plus the number of trays, samples can run continuously without human intervention. With regards to the widest separation range of our gels, we have 17 different gel kits for analysis, and they are specially designed for looking at different types of separations. We can separate DNA fragments from very small DNA fragments in the 300 base pair range with just 3 base pair resolution to very large DNA fragments through 20,000 base pairs. Some of our gels can actually separate in under 15 minutes if desired. We have gels for next-gen sequencing fragment analysis. Both the typical size range of the Illumina and the Ion Torrent instruments, as well as large fragment smears like you might use on a PacBio instrument or a nanopore sequencer. Our instrument is the only instrument that has gels which can separate fragments through 100,000 base pairs and look at smears through 15,000 base pairs. We can look at incoming raw materials like RNA and genomic DNA. We were the first company to automate the quality and quantity analysis of genomic DNA and the first to introduce a genomic quality number. In addition to our kits for looking at total RNA, messenger RNA, and ribodepleted RNA, we are now the second company to have a microRNA, small RNA reagent kit available for use. If you're interested in CRISPR or are doing tilling, we have reagent kits that can automatically analyze your heteroduplex cleave fragments typical of the CRISPR-Cas9 gene editing events. And lastly, we have gels for looking at plasmids, both linear and supercoiled plasmid DNAs. All of our kits work virtually the same. You simply take dye, add it to your gel, put it on the instrument. You put buffers into buffer trays and load them onto the instrument. And then, simply taking 2 microliters of your sample plus 22 microliters of our diluent or diluent marker solution and load it onto the instrument, within 7 clicks of the mouse, your samples are off and running and you have nothing more to do. If you want to load more samples, as I said, our sample trays are always available and accessible for loading samples, and those samples can be queued and placed onto the instrument for continuous operation. With regards to flexibility and dependability, as I said, the array stays in the instrument. No one needs to handle the array in between runs or on a daily basis. In fact, after every run, our capillaries move to a homing position, and that homing position contains a capillary storage solution which protects the capillaries and keeps them ready for optimum performance. The arrays are robust. They are not fragile. They are used for thousands of injections and are typically only handled twice, once when you put it in the instrument and once when you take it out of the instrument. Now, as I said, the gel is loaded automatically, and this typically leads to a negligible failure rate because we rarely have improper loading of our gel. Our customers tell us that we typically have no problems with capillary clogging, and gels can be held onto the instrument for an extended period of time. For our qualitative gel kits, they can be held on the instrument for up to 14 days, which means you can load the instrument and then simply set your samples on the instrument and run them with continuous operation. One other feature of our instrument is the ability to change the run conditions during the electrophoresis. Both the time and the voltage can be changed if desired for the electrophoresis. Lastly, with regards to our software, we have talked to our customers and revamped our software, adding new features. Some of those features are listed here. We can now do batch processing of samples. From the very addition of the first sample put in, we can generate a report without any intervention. We are working on LIMS integration with Genologics for data acquisition and data documentation. For customers that have many fragments and have many samples with complex fragments, we've created an advanced flagging feature that allows reports to be generated and allows individual capillaries to have specific sample sizes and different from one capillary to the next. As I mentioned, we are working with CRISPR-Cas9, and we have a plugin that allows mutation frequency analysis of pooled samples. Our genomic quality number is a user-defined threshold that allows the users to define what is good quality DNA. This way, a different threshold can be set depending on what type of samples they're working with. Think of formalin-fixed DNA compared to whole genomic DNA from fresh tissue. Big difference in the sizes. And lastly, our RNA quality number has been tested and retested with hundreds of RNA samples, and it matches very well to the legacy method. But you know what? Don't take our word for it. These are some of the things that our customers are saying about our instrument. So the fragment analyzer is the premier instrument for automating the analysis of nucleic acid fragment and smears. and does have unmatched features and benefits to improve workflow in the laboratory. We have sold instruments around the world to top quality organizations, research institutes, and corporations, and have a very strong relationship with all of the NGS platform players, including Illumina, PacBio, and Roche. So I would encourage you to come to our booth, number 53, right over here, or go to our website to learn more about the Fragment Analyzer, and then request a demo to find out why our instrument is the premier instrument for automating fragment analysis. And with that, thank you for your time.